Hello, this is Dr. Miller. As you know, I am in Chicago at the AMSA board meeting. So I appreciate you coming in and listening to this lecture. I'm going to cut this lecture a little bit short because, as you know, our colleagues from Tyson Foods is going to be here to talk to you about them as a company and also intern and career opportunities there. So please take advantage of that. I encourage you strongly. Today's lecture is about conversion of muscle to meat. And I put the sources up here from your textbook. We'll be using information from Chapter 4 and Chapter 5. But the main objectives of this lecture are to understand how mechanisms of homeostasis induce physiological changes post-mortem, and to understand the steps of rigor and the physiological changes in muscle that are a result of rigor mortis. One of the things that I find fascinating about rigor mortis is we know a lot about conversion of muscle to meat, but there are a lot of things that happen during this process that we still haven't been able to fully understand. And when we go back and look at the effects of pre- and post-harvest factors that affect meat quality, there's always this big hole of variation not explained that's associated with slaughter day. And what that means is that the day the animal is slaughtered, the conditions, whatever happened, whether that's cooling temperature, time, stress, the animal, there are lots of factors that affect that. But when we control for everything that we can for variation, there's still the largest amount of variation in quality that's associated with slaughter day. That means conversion of muscle to meat is very important. Well, conversion of muscle to meat, of course, is where we're going to convert the live animal or live muscle cell to a meat muscle fiber. And all of the mechanisms that are going to, or the changes that are going to have during this conversion process, are driven by homeostasis. And as remember the age-old definition of homeostasis, you should know this, is the maintenance of a physiologically balanced internal environment. We talked in the last lecture about contraction, what's going on with contraction and relaxation. And we know that this is a very delicate, balanced system, and that there are mechanisms put in place to keep this system where it needs. We need to have ATP produced. We need to be able to have all the systems working. And there are actually a series of checks and balances that we haven't talked about in great detail within the biological system to keep everything functioning. And why we have homeostasis, part of it is to provide a means for coping with stress. And when I say stress, I don't mean that stress necessarily that we put on animals, but environmental stress, anything that's going to be changing their internal environment, we would consider stress. But that muscle fiber wants to keep that muscle fiber in a very narrow range of physiological conditions. It wants to be in a very narrow range for pH. When pH starts to be too higher or lower, and physiological pH is 7.4, we know that things start to fall apart. Enzymes don't work as effectively. Membranes start to leak. Everything starts to fall apart. So also temperature plays a big role in that. And if we change body temperature, if you've ever had a fever, you know what that does to your energy supply. If you've ever been too cold, you also know how that slows everything down. So we need things to function at optimal pH, optimal temperature, and oxygen concentration is also part of homeostasis. We need that mitochondria functioning. We need to be able to produce the ATP so that we can keep up that energy supply. The homeostatic mechanisms in the body, they are presided over by the nervous system and endocrine system. The nervous system, which sends signals that says fight or flight, and the endocrine system, which if we're going to fight or flight, going to produce release hormones that are going to regulate rate of metabolism and a number of things. So these, the nervous system and the endocrine system, they serve to communicate and trigger mechanisms that that coordinate or adjust the function of various organs during stress. So a lot of things that happen during stress. If that stress can be me going out and deciding to run even a 10K at this point in my life would provide a lot of stress to my system. Stress can also be being scared, being frightened, anything like that. We're going to relate homeostatic change to the conversion process. So those conditions in the immediate pre-slaughter period can alter post-mortem changes that affect meat quality. You can see that if we start the homeostatic system too soon, we're going to have hormone release, we're going to have fight or flight system stimulated, and that's going to change the physiological environment of that muscle cell. We'll come back and talk about that bullet a little bit. We're not going to really talk about it. We're going to talk about it when we talk about pork quality and when we talk about pre-harvest factors that affect meat quality. So don't forget that. But basically what we are interested in this conversion process is the loss of homeostasis. We lose control of the system within that muscle fiber. Those factors, the nervous system is going to be lost within 4 to 6 minutes after bleeding. So the nervous system, which oversees homeostasis and allows the biology of that muscle fiber to adjust, is going to be lost. And we're also going to lose the system that protects against microbial invasion. The lymphatic system is going to stop working, which is also part of our immune system. We're going to no longer have white blood cells circulating. We're also not going to have any blood circulating. So we're going to have loss of the ability to bring in food or glucose, and we're going to lose the ability to take waste products out. So we basically strand this muscle, this cell out there to be on its own, and eventually it runs out of resources. And also, when we go through this process, we're going to have some altered membrane properties, which is going to have an influence on how rapidly things go. So homeostasis is very important to us in the living animal. What are we going to do from a practical standpoint when we do conversion? What I want to do now, after kind of talking about homeostasis and kind of planning that in your mind as far as what's going to drive some of these changes, is I'm going to look at what we actually do for conversion of muscle to meat on a hands-on basis, and then start talking about getting down deeper into the muscle fiber, and end this lecture with individual components that are changing. Well, when we look at conversion on a practical basis from an animal, from muscle to meat, the first conversion step is we immobilize animals. Now, there are some species like poultry that don't require immobilization. There are also animals like kosher slaughter, special regulation animals that are not immobilized. For commodity beef, pork, and lamb production, we immobilize. And that is because of the Humane Slaughter Act, saying that we are going to convert this animal into meat in a humane manner and be as humane as possible. So what is the first step in this is immobilization. And immobilization is where the animal is going to basically be rendered unconscious. They're to sensibility. And it's conducted prior to bleeding. We can apply different types, but we want the animal not to know what's about ready to occur. We don't want them stressed prior to this. We don't want any discomfort. We want this to be a very rapid process so that they don't suffer. The types of immobilization techniques used. Electrical, with about not probably less than half the hogs today are stunned with electrical stunning. Some poultry, fish, also some turkey are use electrical stunning. A lot of poultry does not use any stunning. And in electrical stunning, they'll use 70 to 90 volts, about 3 amps, for 2 to 10 seconds. And I don't know if you've ever had an electrical shock, but this is enough to render the animal unconscious. And then we can go to the next step, which is going to be bleeding. We also have, especially in beef cattle and in sheep, we mainly use concussion. Either the poleaxe or the captive bolt, and that's what this is a picture of. I know this is my least favorite part of meat science, I'm sure most of yours, but in a concussive type of system, we're rendering the animal unconscious by applying some type of concussive force to the head cavity. And so they basically have a concussion. In swine, about 60 to 70% of swine now, and it might be a little bit higher today actually, are immobilized with CO2. So the animals are lowered in groups down into a CO2 chamber where they basically fall asleep because they don't have sufficient oxygen. And with kosher kill, there's no immobilization, and we have more quality control problems with kosher killing meat than we do with regular meat associated with the immobilization step. And we do have one major quality issue associated with immobilization. If the animal is immobilized and doesn't go into full unconsciousness and has some awareness that causes stress, the animal will most likely panic, and we will see the fight or flight system, the hormone control, the nerve system stimulate fight or flight. And what happens is that we end up with what we call fiery fat or ecchymosis. And shows ecchymosis here. Fiery fat looks like you shot it with a BB and that there's like a little blood lanes from the capillaries there, but I don't know how well you can see this. You should see this on your individual slides on eCampus, but it looks like this meat has measles. With improper immobilization, the animal's going to go through short-term excitement. It's going to cause them to overheat. Usually this is caused by rough handling or improper training of employees in how to immobilize. And the result of that is you're going to have increased blood flow to the capillaries and blood pressure increases. You also have at the same time vasodilation, and the capillaries burst because there's higher pressure, there's more dilation, there's more blood flow. And we know that this occurs when the animal its heart is still pumping because it's related to blood flow to the capillaries. And we don't get any blood drainage post-mortem. One of its common names is called blood splash. People will look at this, and obviously there's nothing wrong with eating this, but visually it's unappealing. And when we have things that are going to have higher levels of microorganisms in the product, then we're also going to have issues with more blood. We're going to have more issues with microbial growth. So ecchymosis or fiery fat, that is associated with a quality defect if we do not have improper immobilization. The next step of the conversion of muscle to meat from a practical standpoint is exsanguination. And exsanguination is where we remove the blood from the animal. The target here is to remove about 50% of the blood. If we remove less than that, we're going to see darker colored lean that's not going to have the same flavor profile. In cattle and sheep, we exsanguinate by cutting the carotid artery and the jugular vein, and in swine, we'll go down into the chest and cut the anterior vena cava. The issue with both of these is making sure that we get a proper cut to have as much blood removal as possible. What happens when these major arteries and veins are cut is that blood pressure is going to drop, and so we're going to get initially a lot of blood removal, but those capillaries are going to constrict, and it's going to be hard to get some of the blood out of the extremities of the animal. Also, the circulatory system adjusts by increasing pumping, and so the peripheral vessels will constrict to maintain their pressure and hold blood in the vital organs. And one of the reasons we worry about how much blood we get removed from the animal during this process is that blood is an excellent medium for microbial growth. So if we have improper exsanguination, we're going to have meat. The resultant meat is going to have a lower shelf life, and it's also going to look darker. So my story for this part, for exsanguination, I had, I don't know if it was a pleasure, but I was part of a team that developed ostrich slaughter and fabrication procedures for the ostrich industry in the mid-1990s. And the first time that we went to harvest an ostrich, we were kind of like, what do we do? So we used captive bolt, and then we exsanguinated like we would sheep, up right underneath the head of the animal. Brought the meat back, and we weighed the animal, we weighed the blood, and they brought the meat back, and we asked our colleague, who was Dr. Alice Blue McClendon over in the vet school, how much blood does an ostrich have? Because we didn't get very much blood out here, because as we were bleeding the animal, it had to come down the long neck of the ostrich. And we found out that they have about the same percentage of blood as other mammals or other avian species. And so it's like, okay, I think that we didn't get good blood removal. When we first ate that meat, I can tell you it was incredibly serumy, bloody. It had a lot of metallic and liver flavor. And in fact, my graduate students played a joke on me because they know I don't like liver, and they gave me the meat without telling me what it was, and I immediately spit it out because it was so liver-like to me. So it's very important that exsanguination is done properly, and one of the things that we do in during this process is a lot of people will use some type of low voltage electrical stimulation to increase the amount of blood removal during exsanguination. One of the other reasons that we want to eliminate blood is that when we eliminate blood, this is really the step that causes death. And we've eliminated the transport of nutrients to the muscle and the removal of those waste products. And if we have that improper blood removal, we are going to have a shorter shelf life and a darker color, and the flavor changes are those associated with metallic and liver and serumy flavors, and we know that those tend to be flavors that consumers do not like. And so then what happens for the rest of the conversion? One slide. Remaining steps. We remove the feet, the head, the hide, the tail. We complete removal except for kidneys, the viscera and the plunk, and wash and apply during this process any antimicrobial steps or microbial interventions, and then immediately refrigerate. When I first came back to A&M from industry, the average time for conversion on the slaughter floor was about 22 minutes. Now it's about 45 minutes because of interventions from a food safety standpoint. So within 45 minutes, animals are converted into carcasses. And then that starts the conversion process, but the conversion process from a biochemical standpoint continues for until that meat is cooked and consumed, basically. So let's look at kind of what's going on. And I like this slide. I told you I like this slide on Thursday because I want to come back and use it for the conversion process. We're going to go down another step and look at what's going on. We know kind of the steps of converting from a practical standpoint, but let's start looking at what's going on in that muscle and inside that animal. We call this process the process of rigor mortis, and rigor mortis stands for stiffness of death. And so basically what we're going to do is the animal, when we start the process of conversion, the muscles are very flaccid and they move very easily. And during the onset of rigor and as we go through the process, the muscles basically go into full contraction, which is called tetany, and they go into contraction and they can't relax. So keep that in mind, that that stiffness that develops after death is a loss of extensibility or elasticity of the muscles. And what that is from a practical standpoint is we're getting contraction of muscles and inability to relax the muscles. So think about that. Let's also talk now about some of the physiological changes that happen in muscle as they try to maintain homeostasis. Remember those muscle fibers are in there, and they're going to try to stay alive and keep those very narrow range of homeostatic systems in there, the same pH, the same temperature. So the muscle is going to try to maintain energy supplies. So as it goes through glycolysis, we find that the muscles become unextensible because they lose the ability to produce ATP. When we have exsanguination and circulatory failure, we know that the blood-borne oxygen supply to the muscles is going to be limited. And there's only enough oxygen stored in a muscle for a few twitches. So what's going to happen? This system, the system trying to produce ATP and maintain ATP levels, is going to go from aerobic metabolism to anaerobic. So we're going to start being dependent on glycolysis. But the issue here is we no longer have the circulatory system to carry nutrients or waste products away. So as this muscle goes from aerobic to anaerobic, we see the buildup of lactic acid because pyruvate's there with the hydrogen ions, and they're out there in the sarcoplasm. Lactate dehydrogenase is functioning. It ties up some of those hydrogen ions and forms lactate so that lactate can be transported out of the muscle. Oh no, can't take it away. So lactic acid is going to build up, hydrogen ions also build up, and we have a drop in pH. Also, the line of communication between the muscle and its external environment is lost. So there's no communication, no nerve impulse, no ability to do for contraction. And because of these things, where this muscle's trying to maintain that very narrow balance of physiological conditions, we end up using up all of our oxygen, and the muscle eventually loses the ability to maintain ATP. And if we don't have ATP, then the muscle, the heads of the myosin can't release to an actin binding site. That's the most critical part. So we always talk about conversion in the living animal. What we're really thinking about is the conversion from aerobic to anaerobic metabolism. And we know that oxygen is picked up in the lungs, carried to the muscle by the hemoglobin pigment, And then myoglobin, which has a higher affinity for oxygen than does hemoglobin, picks up the oxygen from the blood system. The muscle stores of oxygen, they're sufficient to support aerobic metabolism only for a very short period of time. So as the stored oxygen is depleted, and we can't get any more, the aerobic pathway through the citric acid cycle and the cytochrome chain stops to function. And then that energy metabolism is shifted to anaerobic pathway, and lactic acid accumulates in the muscle. So this is a review of what I said with the last slide, but hopefully this helps you to see those steps a little bit clearer and to be able to refer back to them. So what happens in the muscle? What are the post-harvest changes? The depletion of oxygen that causes those muscles to revert to anaerobic metabolism and the production of lactic acid, then that causes that increase in lactic acid and a decline in pH occurs. And this is the most significant event in postmortem muscle. And we monitor how rigor is occurring by using muscle pH. That's not only a good practical way, because we have pH meters that are almost instantaneous reads, not quite, but close. And we know that that change in pH is indicative of the change from aerobic to anaerobic. Think about that, because we're really measuring lactic acid buildup, which is the inverse of pH concentration. The rate and extent of that pH decline is going to be dependent on a lot of things. I want you right now to set in your mind that every muscle and every location within a muscle will have a slightly different pH decline. What does that mean? And what I want, the perception I want you, or the concept I want you to get, is that when you sit and look at a carcass in a cooler, and you look up at the round, and you see one of those muscles, the outside of that round is cooling faster than the inside, so there's a temperature gradient, and temperature affects how rapidly enzymes work. So it affects metabolism, which is going to affect glycolysis, which is going to affect pH decline. So within the animal, within a carcass, within muscles, the process of conversion is going on at different rates. And we're going to talk more about that. And some of that rate is going to be dependent on how much glycogen is available in the muscle, and also postmortem temperature. So what temperature is in the cooler? We're going to talk a little bit. I have one paper on chilling rates, and that's because of this effect. It's going to be dependent on ATP concentration, what's initially there, and then how much that animal has the ability to produce based on the glycogen content. And temperature throughout the process is going to affect the rate of these reactions. And also, this whole process is going to be dependent on calcium concentration. We know that within post-harvest changes, we'll see a normal pH decline is usually gradual, and it'll go from physiological pH of 7.4 down to about 5.6, 5.7. It can go down to 5.2. Actually, we've gotten pork carcasses down to 4.7, 4.8. The pH change, it can be very slow during the first hour, and it can even remain relatively high with time. It can drop rapidly very early on. It can be someplace in between. So that pH drop is not consistent across all muscles and across all animals, and how rigor proceeds is dependent on that pH. Well, there are negative effects associated with all these changes. The first negative effect I want to talk about is when we have with blood removal, we lose an important temperature control. Remember that picture that I like that has the muscle and the whole systems in there? We talked about how the blood system takes heat out, and that's dissipated through the lungs and through the skin. So if we lose the ability to dissipate heat, it means those exothermic reactions are going to cause a temperature increase in the muscle post-mortem. And so, and immediately post-mortem, you're actually going to see inside a muscle, especially inside a very deep muscle, an increase in muscle temperature. The heat is generated by ongoing metabolism, and the sooner we get that carcass into a cooler and get the heat out of there, the better off we are from a rate of rigor issue. The rise in the temperature is going to be affected by the size and location of the muscle. The biceps femoris muscle is the largest muscle in the animal. It's the outside round along, and so as that carcass is hanging, it's on the outside round. It attaches on the inside. It's close to the bone, the femur bone, and on the inside of that muscle there, and also on the inside round, which is on the opposite side, we see where the muscle attaches to that bone. That is the location, the carcass, that chills the slowest. And so we're going to have a much different pH decline and temperature decline inside the muscle than on the outside of the muscle. And I have a lot of data from a company in Mexico where we monitored location in the cooler. We looked at different size carcasses, and because they were having some color issues with that inside round and outside round. And when I looked at it, what was happening is they're getting discoloration where the bone was after they pulled the muscle and cut it. It's like, that's really obvious. That's a pH effect. It's a temperature decline effect, because you're dealing with the part of the carcass that chills the slowest. So you're going to have a little bit lower pH because rigor has a chance to go longer. Also, if you have animals that have more fat covering, they're going to chill less rapidly, and you're going to have heat be able to be maintained within the muscle. And we know fat is an insulator. Think about whales and walruses and seals that have fat to keep them warm inside. If we have more fat covering, we're going to have more issues with getting heat out of the carcass. Ambient temperature in the slaughter area is going to also affect the temperature control. When it's 105 out, the temperature on the slaughter floor is very high, and that because of that, the carcass is not going to be able to dissipate heat, but it's going to maintain temperature before it gets into the cooler. So I said the average time on the slaughter floor is about 45 minutes. If in the middle of summer the chain stops in the slaughter floor and carcasses are out there an hour and 15 minutes, I can tell you that they've already partially gone through very early, not the first step of rigor, and they're well into the second step by the time they get put in the cooler. And you're going to have a very rapid pH decline in those animals because all their metabolism is going very rapidly. The length of the slaughter operation, if that relates to breaking the chain down, but even if anytime you delay getting that carcass into the cooler, you slow it down, it's going to affect how rapidly rigor mortis sets in, and the temperature in the chill cooler. Today, most chill coolers are going to have spray chill. They're going to use water to try to help keep evaporation loss minimized in the carcasses so that they don't have any carcass shrink, but water also helps in transferring temperature. So a moist environment makes it feel colder, helps dissipate heat from the carcass more rapidly. And we're going to talk about some of these things when we get to the Sable article. The other negative effect that we want to talk about, other than temperature control, is low ultimate pH coupled with high muscle temperature. And so if we have high temperature, we're going to have more rapid pH decline, and that pH decline has the ability to go further than a normal muscle because you don't have any temperature limitations. And you can with that high muscle temperature, you can also get some protein denaturation. Heat denatures proteins, and when you have protein denaturation, those proteins lose some of their protein solubility, some of their functionability. They lose their ability to hold water, and they also, in further processing, have some protein binding capacity issues. We'll also see in animals that have low pH at high temperature, we'll see some loss of intensity of the color muscle pigment because the main muscle pigment is myoglobin, and myoglobin is going to denature as well. We're also going to be associated with increased free water is going to affect color. And we're going to talk about those things later in the semester. So, in this whole process, a lot of negative things are happening. We are basically stranding this muscle out there to be dependent on its current food supplies, oxygen in this case, glycogen as well. You should know that most muscles have enough glycogen in them that pH could drive down to about 4.5, 4.6 in that range, even down to 4.2 in some animals. But we don't see all glycogen being depleted during the process of rigor. Muscles have about 1% glycogen that's left after the process of rigor mortis on an average. So we've stranded the muscle, but most importantly, we've removed its circulatory system, so we can't remove any byproducts of metabolism. The most important byproduct is lactic acid, and we see that pH decline occur as an indication that the muscle's starting to struggle. And as that lactic acid builds up, due to the limit of the mitochondrial function, which is limited by oxygen level, we see the conversion of aerobic to anaerobic metabolism. We see less ATP. We eventually run out of ATP because anaerobic metabolism isn't as efficient. We also lose ATP. It cannot complex with magnesium. So not only do we have the myosin heads stuck because there's no ATP to bind to them to get them to release, the magnesium ATP, the oil is gone to allow that sliding of the thin and thick filaments. What's interesting is that in normal contraction of a muscle, we'll have from 10 to 20% of the myosin, actin myosin cross bridges formed. During the process of rigor, we have almost all the actin myosin cross bridges are eventually formed. So this is just like massive contraction with no ability to relax. That's all driven because we lack the ability to produce ATP, and those myosin heads cannot not release. So what I want to do now is I want to go ahead and start talking about what's really going on. We talk about rigor mortis in 4 phases. We talk about the delay phase, onset phase, completion, and resolution. Each of these 4 phases are defined by the elasticity of the muscle. So we defined elasticity as the extensibility, how the muscle has ability to stretch and relax. That scale is based on, extensibility scale is based on elastic to stiff. So lower is very elastic, very pliable, and stiff, of course, is very rigid. What we find is that in the delay phase of rigor, the muscle is very elastic, really no different than it was prior to starting the process of conversion. But eventually that muscle, fairly soon on, starts to slowly lose its extensibility, its elasticity. And that's a pretty linear effect. We call that phase onset. Once onset is completed, notice I use that word because it goes into the completion phase, and there's no change in extensibility, but the muscle is very stiff at this point in time. After the completion phase, we see that there will gain some extensibility, some softness in the muscle, and that is called resolution of rigor. Basically, completion occurs in the first 24 to 48 hours, and then we start the resolution phase by usually 3 days. So let's talk about what's really going to go on in each of these steps or each of these phases, and I'm going to keep the extensibility line up there. So first let's talk about the delay phase. Let's think about what's going on here. This is the time during which the muscle is very elastic, and that means that there's still ATP available. The muscle's still able to relax. From a tenderness standpoint, this is defined as usually the most tender meat. Pre-rigor meat is very tender. So everything is, there's no rate limiting step, which means that we're in aerobic metabolism. Creatine kinase is used up early on in this system, the short-term energy supply, but we have enough oxygen that we can keep the mitochondria functioning. So we're going to use up creatine phosphate in this phase because we're going to have ADP produced, and remember that reaction equilibrium lies very heavily towards production of ATP. Glycolysis is going to be going on, but we're going to be using up some oxygen as the TCA cycle and cytochrome chains still function. And so if we look at this for delay phase, we see starting at the bottom, mitochondria functions fine, isn't it? During delay, it's still able to go. Well, it's still able to go because look at the oxygen concentration. Yes, we're using oxygen up during delay phase, but we haven't come to the point where it's rate limiting. So delay phase uses up oxygen, but we still have sufficient oxygen for everything to work. Glycolysis is working fine because there's nothing to really limit it. We are using up glycogen because that's going to be our energy store. We have to go to the cellar and use up whatever we have there. So we're going to be getting glucose not from the blood system here, but from glycogen. And ATP is able to be maintained at a very steady pace because all the systems are in place. However, creatine phosphate is going to decline very rapidly, and in fact, we use up all the creatine phosphate in the delay phase. Well, everything's going along fine, but now we start to have some issues because we're going to start having some rate limiting factors occur. What's the biggest rate limiting factor? It's going to be oxygen. So we see the muscle, remember in onset we start losing the extensibility. We've used up creatine phosphate by this phase, so there's none left. It's hard to measure creatine phosphate levels early post-mortem, it gets used so rapidly. And from the glycogen stores, they're going to continue to be used, but they're really not rate limiting at this point in time, but they're being depleted. But we see oxygen becomes the rate limiting factor. We're still going to be using up oxygen. We still have some oxygen in this muscle, but not enough to maintain full mitochondrial function. So slowly, in the onset phase, we lose mitochondrial function. And if we lose mitochondrial function, this muscle fiber is more dependent on glycolysis for ATP production. And so glycolysis is able to continue to run, but it's going to eventually limit glycolysis functioning. So we see not as much ATP is being produced. We slowly shut down ATP production, because as we shut down the mitochondria, it doesn't just go from being turned on to being turned off. It slows down until it eventually almost totally shuts off. And we can see that as ATP is decreasing in its ability or in its concentration level, we start to see a buildup of lactic acid. Lactic acid is an indication of aerobic to anaerobic metabolic conversion. And as that lactic acid is building up, the pH starts declining. We continue to see glycogen concentration decrease. And we see some actomyosin bridges form. And as we decrease ATP concentration, we get more and more actomyosin bridges forming that can't break apart, because they need ATP to do that. So if we go back to our chart here, we can see that, let's just go down to the bottom here again. We know that we're going to lose extensibility, because that loss is due to the myosin heads binding to the actomyosin binding site, contracting, and not being able to relax, right? No ATP to relax. There's no ATP to pull calcium out of the sarcoplasm, to stop, to initiate relaxation, and there's no ATP to activate the myosin heads to allow them to relax. So look at the oxygen supply here. The oxygen supply is in this dark black dashed line. Now we've used oxygen in delay phase, but notice that we continue to decrease the level of oxygen. But we are now in onset, which means that oxygen level starting to be limiting, and it's limiting mitochondrial function. See how mitochondrial function just slowly declines? As that mitochondrion uses the oxygen within that muscle cell or muscle fiber, it starts to shut the mitochondria down. Glycolysis is still going, but the interesting thing about glycolysis is it slowly slows down. And it isn't ATP, lack of ATP, that's causing glycolysis to slow down. What's causing glycolysis to slow down is the increase in lactic acid concentration, which is decreasing the pH. Because remember, glycolysis is 12 enzymes. Each of those enzymes have a optimal pH and temperature that they function. We call it enzyme kinetics. When I learned about rigor mortis, I all of a sudden understood why I needed to know about enzyme kinetics. Because when pH decreases, those enzymes are going to decrease their functionality. Now they're still functional, but they get down to only partial percentage of their ability to continue to work. So what we do is we slow down the rate of glycolysis as we increase pH. Glycogen continues to decline. We never use all of the glycogen, not really. There are probably a few exceptions to that, but we know that we still have some reserves. ATP is declining in concentration of ATP because we know mitochondria have been shut down eventually, and then as rate of glycolysis slows down, we're going to slow down how much ATP is produced because we have no creatine phosphate anymore. This whole system is going to be dependent on those three enzyme systems involved in aerobic metabolism. Now, as we see, as we proceed down and we shut glycolysis off, we're going to be more dependent on glycolysis for ATP production, and it doesn't produce as many ATPs. Remember, 3 versus 37. So we see during that time, we see lactic acid buildup, which is inversely related to pH decline. So pH is declining. When we're monitoring pH and temperature immediately post-mortem for the first 24 or 48 hours, what we're measuring is how fast this is going. How fast it's going and how long it's going, and what is that pH decline. And then we get into completion phase. And completion is the third phase. And we get 90%, some textbooks say 100% of available actin myosin bonds are formed. In other words, thick filaments and thin filaments are bound together, and they are there, and they are stuck. We know that there's no ATP can no longer be formed, so we can't unstick the myosin heads. There's no ATP to activate the myosin heads. There's no ATP to complex with magnesium to allow that oil and that slippage back. Creatine phosphate's gone. It was gone, really it's gone early in delay phase. We have a very low glycogen concentration, but there's still a little bit left, but we have very low ATP. If we don't have ATP, nothing can really work. The pH drop is almost or is completed by completion, and we have very high lactic acid concentration. So everything just slows down and comes to a standstill, and that muscle becomes inextensible. So in the completion phase, this is the easy one because all the lines are parallel, all right? No changes. Everything's done. And then we go into resolution. Resolution is called postmortem aging, softening of rigor. Those are the common names for it. Basically what's happening during resolution, we don't see any change in ATP and the things we've been talking about, but we do see changes in protein, and it's really those softening that we see is protein degradation. And that protein degradation is in the major structural proteins found in that muscle fiber. Now we have, because we've had long-term hormonal control and tissue metabolism is stopped, and temperature is now fallen, then the meat temperature should be maintained at very low temperatures. We know that fat is going to go from being somewhat fluid and liquid at body temperature to solid at cold temperatures, and we've lost the antioxidant control mechanisms, mainly vitamin K in the muscle, and so now this muscle is this cell is now susceptible to lipid oxidation, and the I'm going to skip that for a minute because we're going to talk about that later. And beef and lamb, rigor mortis basically gets to the completion phase. It depends on temperature and area and whether they're using electrical stimulation. But 6 to 12 hours, it could be up to 24. And pork will go through rigor mortis in 15 minutes. In extreme cases, that's not a good thing, up to 3 hours and even 6 hours. Usually we take final pH at 24 hours is what most people consider to be ultimate pH. So what's happening during resolution, where we know that most of the improvement, even though it's not as grade is what we saw in the delay phase is due to structural proteins degrading. But what we're going to see is that pH is actually drops a little bit during resolution and storage, but that's due to microbial growth. In vacuum packaged meat, the main microorganism that's responsible for spoilage are lactobacillus organisms. They produce lactic acid. We do see some increase in lactic acid, which is affecting this pH decline. That's from microbial growth. Creatine phosphate, it's been gone since the delay phase, so of course we don't see action there. We don't see the ability to produce increased concentration of ATP or glycogen. Rate of glycolysis and mitochondrial function are pretty well flat, and oxygen is no longer able to come into the muscle and be present. Now, I do need to qualify that. If you have meat that's on the surface exposed to air, like an aerobic packaging systems, like the trays with the PVC overwrap, you will see some mitochondria function because exposure to oxygen will turn that on, but it has minimal effect. So rigor mortis is, well, this looks like it's kind of complicated. I walk through this in a gradual basis to try to help you put together what's going on, and I start from the big and go down into the biochemistry of what's going on so that hopefully you can tie all those concepts together. But as I said before, I can't overemphasize variation in rigor mortis across muscles and across animals. pH decline is not static. I showed you one pH decline on one chart. That's not always representative of what pH decline is going to be occurring in actuality within that muscle cell, because red and white fiber types also affect pH decline. Temperature, as you know, pre-harvest slaughter excitement or stress can affect it. And so we have a lot of muscle-to-muscle variation as well as animal-animal variation in onset of rigor. And some of the, we get a rapid onset of rigor in some of the conditions, and this is the resultant meat of rapid onset of rigor. In this case, this animal was very susceptible to pre-harvest stress, and so the fight or flight system was able to kick in, and we had metabolism was running at very high speed, and pH declined very rapidly. This is the 15-minute rigor I was talking about in pork. We can also have animals that have a very high pH because they've been stressed for a long time. Again, we'll talk about this in more detail later, just trying to set the groundwork here. And so they don't have very much glycogen, and they will have ATP will be utilized very fast, and they don't have any glycogen to convert ATP, so they end up with very little pH decline. And we have all variations between that. And we're going to talk a little bit more about the practical effect of going through rigor, and the most important thing, not only what's going on from a physiological standpoint, is understanding that pH is our way of measuring this. So in summary, what I've talked about today, I started out talking about homeostasis and homeostatic regulation in the physiological environment. And then we talked about the steps of rigor from a practical standpoint. And then we moved down into understanding the physiological changes. And I told you I would come back to this slide because I think you can see what's going on, what's going to happen here when we have exsanguination. We cut off the ability to take out nutrients, and we cut off the ability to bring nutrients in. So this muscle cell is stranded. And so we can have glycolysis occur, but once we have limit in oxygen level, we shut down the mitochondria, which is the TCA cycle and electron transport. So we only get 3 ATPs produced. And that slows everything down. We also can't get lactic acid out, so we start losing the ability to generate ATP. Those muscle myosin heads are going to get stuck. Some of them, not all of them. It's going to continue to try to use ATP up and get everything done. It's going to keep trying to run glycolysis, but as lactic acid builds up, the byproduct of glycolysis, it decreases the pH, and those 12 enzymes responsible for glycolysis lose their ability to function as well. So that's kind of what's happening in rigor mortis. I think this kind of gives you ability to look at what's going on and walk through that. Hopefully the charts and the way that I talked about the charts will help you. I'm going to end this right now. Thank you very much. If you have any questions, I'll be back on Thursday, and I look forward to working with you and have fun with Tanner and Bill. Goodbye.